When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential LAMP amplification. This LAMP dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatemers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
have also very promising results in this new method, which will be much more convenient and much more cheaper for the old governments who are dealing with the disease.
You don't have to be in the lab, it can be at home. It can be in airports, it can be in theaters, it can be workplaces. It's actually it can be in every place that you want to do some kind of fast screening of the public in order to make sure that everybody that goes into the premises is actually healthy and doesn't have the corona.
The large S glycoproteins are used by the virus to gain entry to human cells. They most likely attach to angiotensin-converting enzyme 2 receptors on the cell membrane, allowing the virus entry. The exact mechanism for this is not known. Most likely, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic material, a single-stranded RNA. The virus hijacks the cell's machinery to replicate the RNA in N proteins and uses the endoplasmic reticulum to form its M protein outer layer and the all-important S protein. After replication, the virus is carried by the Golgi bodies out of the cell in a process known as exocytosis, so that it can infect other cells. Meanwhile, the stress of viral production on the endoplasmic reticulum eventually leads to apoptosis, or cell death.